A very good morning to one and all. What a tiny single minute virus which cannot even seen through the naked eyes can cause havoc throughout the world, how it can shape this world. We are all experiencing that in this current pandemic. So today we'll be discussing the practical aspects of virology. Myself, Dr. Vishal Kulkarni. I am Associate Professor in the Department of Microbiology. So let's get started with the practical aspects of virology. Now, what is basically virus? Now, most of the general properties, lab diagnosis, treatment that has been taught to all of you, the students, in the theory lecture in details. Will be just take a quick recap. What do you mean by virus? Basically, what is the virus? Now, the virus is present in the twilight zone between the living and non-living things. Why? The reasons are as below. So basically these are obligate intracellular parasite. Why did I say it is in the twilight zone between the living and non-living thing? Because the living cells like the bacteria, fungus or other parasites, now they synthesize their own nucleic material, their own protein, their own enzymes, so they do not depend on any other host. Now what virus differ in this aspect is they lack the enzymes which are necessary for protein and nucleic acid synthesis. They either contain DNA or RNA, but not both at a time. Now the division, how virus divide in between themselves, it is unlike of that of bacteria. In bacteria we have seen that they divide by binary fission, but in the viruses they multiply by a very complex method. Most of us know that antibiotics which are effective against antibacterial, they are not given in the viral infections. So in viral infections the antibacterial drugs are ineffective, and these are smallest living units in the living things. These are smallest living unit. Their size, it's much smaller than that of bacteria. As we all of know, the bacterial size we measure in the unit of micrometer. Now in the viral size, the size of virus is present in the nanometer or in the angstrom unit. So you can imagine how much it is smaller than bacteria. Now they are classified into DNA or RNA viruses. As I said, they either contain DNA or RNA viruses. We'll be dealing with the viruses that are pathogenic to humans. Now viruses, because viruses that can also affect animals, also plants also. We'll be discussing only those viruses which are pathogenic to the humans. So DNA viruses, among those, these are the groups: pox virus, herpes virus. There are a number of herpes viruses like varicella, cytomegalovirus, Epstein-Barr, etc. Then adenoviruses, papovirus, pyrovirus, and hepatiDNA, which contains hepatitis B virus. So these are important viruses which are pathogenic, DNA viruses which are pathogenic to humans. RNA viruses are more in number, which can cause pathogenesis in human beings, and that includes picornavirus, orthomyxo, paramyxo viruses, flavivirus, rhabdovirus, Rio, and the most popular nowadays, that is most notorious nowadays, not popular actually, the coronavirus. So as I said, these are much smaller than that of bacteria. We measure the size of virus in the nanometers or in the angstrom unit, that is 10 to minus 9 to minus 10. Then among the viruses, the largest is pox virus, that is 300 nanometer in size, and smallest is parvoviruses. Now the virus may be enveloped or non-enveloped. This property, this envelope is made up of generally lipids that is taken from the host cell in which the virus inhabits or it is present. So this lipid envelope is easily destroyed by the disinfectants, the organic chemicals like ether, alcohol, chlorhexidine. So this property is used for prevention of these kind of viral enveloped virus infection. This shapes may be icosahedral or helical. Some of the viruses may contain some extra features on their envelope like papillomas or spike that are present in the influenza virus, even in the coronavirus also. Now, what are the different modalities to do the laboratory diagnosis of this viral infection? Now, all of us know that for lab diagnosis of any kind of organism, we need, what is it required is to demonstrate the whole organism or the part of that organism in the direct sample or direct specimen. So like any other organism, microscopy after sample collection, microscopy is the first modality. Now how it differs from the other bacteria, what kind of microscopy is needed, we'll discuss that in subsequent slide. Then you can demonstrate the antigen of the virus using different kinds of methods like ELISA or modification of ELISA. We'll discuss in detail that also. Then, like bacterial culture, we isolate the virus. We can cultivate the virus using different modalities. And like antigen, we can also demonstrate antibodies in the blood specimen of the affected patient. Now, depending on system involved, the specimen is collected. As we all of know, influenza virus or the coronavirus, in which respiratory system is most importantly involved. So throat swab, nasal aspirate, sputum, these are most important sample in those kind of viruses. Like rotavirus, poliovirus, GI system is affected. So in that case, stool or rectal swab can be collected. The meningitis causing viruses, there are different types of virus which can cause meningitis or encephalitis like CMV. So in that rabies, so in that case CSF, brain biopsy is collected. Skin infections, the virus which can cause skin infection, which can cause papule pustule formation like pox virus, variola, then herpes virus. In that case, you can collect the vesicular or the fluid present in the pustules, or macule papule scraping can be collected. Again, the ophthalmic infection, which are generally caused by herpes virus. In that case, conjunctival scraping is collected. UTI causing virus, where urine is collected. The blood-borne viruses like HIV, hepatitis, where blood is collected. Now this picture you can see, most of us know because of the coronavirus pandemic, where throat swab is collected and it is transported in a tube. That tube is nothing but this tube, which is called as viral transport media, in which the virus remains viable, does not get killed. So sample is collected in this, the swab is collected in this medium. That medium is, if at all possible, immediately transported to the laboratory in a cold chain. So in the transport is done in generally the vaccine carrier, where the temperature is maintained at 4 degrees Celsius. If transport is not possible immediately, it is stored at minus 70 degrees Celsius. Coming to the microscopy now, generally for virus, particularly for demonstration of virus, electron microscopy is needed. But virus which can affect the host cell, virus in which host cell is affected, and during replication of the virus it produces a structure which can be identified using some staining that is easily identified on light microscopy. So that tissues, that structure is called as inclusion bodies. What are inclusion bodies? As I said, these are some aggregates of virus or viral particles which during replication causes some altered staining to that or particularly altered staining to the host cell, so that affected host cell like this here can be identified using light microscopy, and these are called as inclusion bodies. Now there are different types of inclusion bodies. It can be cytoplasmic or nuclear. All these inclusion bodies can be present at both sides, like in cytoplasm and in the nucleus. So classical example of nuclear and inclusion bodies is caused by herpes virus. The cytoplasmic inclusion bodies are caused by pox virus, and the inclusion bodies which are present in the both cytoplasm as well as nucleus is caused by measles. The popular inclusion bodies which can be asked as MCQs, that are Negri bodies caused by rabies, Gurinari bodies caused by pox virus, Zang cells caused by herpes virus. You can see the corresponding pictures here. So as I said, for demonstration of virus itself, we need some modification in the microscopy, which is called as electron microscopy. That setup is not routinely present in every laboratory because it is expensive compared to the light microscopy. But you can see the size and shape of the virus. As you can see, this is hepatitis virus, this is rabies virus, and you must be familiar with this picture, which is a coronavirus. Then another modality in the microscopy which is also available nowadays because of the use of fluorescent dyes, and these dyes which are taken by antigen in some biopsy specimen of the affected tissues. Classical example is brain biopsy in the rabies, where we can demonstrate the antigen in the brain smear. Then, as I said, components of virus can be demonstrated during the lab diagnosis, like the antigen which are detected using some rapid test, like in the HBsAg. Nowadays rapid tests are also available for COVID-19 also. So these are the rapid tests. These are nothing but the immunochromatographic test modification of ELISA. So this is the classical ELISA, where you can detect either antigen or antibodies. Classical example is rotavirus antigen that is demonstrated in the feces. You can demonstrate antibodies also using serum of the patient. Then some other modalities like radioimmunoassay, counter immunoelectrophoresis, immunoperoxidase, and immunofluorescent tests are also available. One which is considered as a dream for every laboratory, the test which is most sensitive, specific, costlier, the infrastructure is required, which is nothing but the PCR, that is polymerase chain reaction, that is detected using some DNA or RNA probe depending on virus in which the DNA or RNA is present. If it is DNA, it is routing the PCR. If it is RNA, the PCR used is a reverse transcriptase. This is the enzyme which convert RNA into DNA first, and then the PCR reaction, polymerase chain reaction, is used. This PCR can be present in the open system, that is open platform system, which require a larger setup, larger infrastructure. Stringent precautions are required while handling these specimen, or closed system like CBNAT, that is Gene Expert, or TrueNAT, which does not require a larger setup, but the disadvantage is having less number of sample can be handled at a time, the capacity of the samples. The cultivation of virus. Now, most importantly here, the difference between bacterial and virus culture is we cannot use inanimate media for viral culture, that which is we used in the bacterial culture, the different types of agar, that cannot be used in the viral culture. So what are the methods for cultivation of the virus? What is embryonated egg method, tissue culture, and animal inoculation method? We'll discuss one by one. starting with the egg method. So this is the embryonated hen's egg, sometimes duck egg can be used. Generally 9 to 12 days old egg is used. These are the parts of eggs. You can see the air sac, then this is chorioallantoic membrane, this is allantoic cavity, this is the yolk sac, then amniotic cavity, and this is the embryo proper. Apart from that, albumin is present in the lower part. Now each site is important where we can inoculate the virus. Different types of viruses can be cultivated into different sites. We can discuss first with the chorioallantoic membrane, where we can inoculate the variola vaccinia viruses. Then allantoic cavity, where allantoic fluid is present, in which we can inoculate influenza, para-influenza virus. This allantoic fluid can be used for production of vaccines, anti-sera. Then yolk sac, where chlamydia, rickettsia can be inoculated. Amniotic fluid, again, in which influenza virus can be inoculated. And so this is the same description which I have been discussing in the last one. Another property of some viruses like influenza virus, which contains some extra structure, by this test in terms of hemagglutination inhibition. So this is hemagglutination and this is hemagglutination inhibition. The chorioallantoic membrane, as I just discussed in the cultivation section, where we can inoculate some viruses like variola vaccinia, which can produce a lesion on the chorioallantoic membrane. You can see here that lesion differ in size and shape according to virus which is getting affected. So these are called as pocks. The lesion are called as pocks, and the counting of pocks are done to approximately convert into the viral count. Plant culture, the small bits of which is more comparatively commonly used comparatively to that of the tissue culture and explant culture, organ culture. So these are the three types of the cell culture that are used: primary cell culture, diploid and continuous cell lines. Now how the cell, what do you mean by cell culture, we'll discuss in the next slide, how it is inoculated in the tube or bottle. So these are the examples of primary cell culture. Then diploid cell culture, that goes into senescence. So single types of cell like fibroblast are used. WI38 and HL8 are classical examples. Some of the cells which are called as malignant cell which have the property of unlimited cell division or indefinite subcultivation can be done using malignant cell. So cancer cells are used like HeLa, Hep2, Vero and McCoy cells. This is the method to form or prepare the cell culture. The tissues are taken that are treated with some enzymes which degrade this tissue and convert it into individual cells. The enzyme used are trypsin. Mechanical shaking is also done. Now these individual product or individual cells are washed and suspended into the growth medium, which contain various nutrients like essential medium, amino acid, vitamin, salt, glucose, some chemicals, fetal calf serum, antibiotics which inhibit the bacterial contamination. Cells are poured or prepared, and in which the sample of the virus is inoculated. After that, cytopathogenic effects are produced that can be seen in the microscopy. So we have to identify whether virus is present or not using these modalities. Like we'll discuss that in one by one electron microscopy, demonstration of antigen, cytopathogenic effect, metabolic inhibition test, and other. We can discuss first the cytopathogenic effect. So in that microscopy, we have to see what changes in the cell lines the virus has caused. So there will be morphological changes if virus is present in the sample. Now those changes will be maybe the alternation of size, shape of the cells, like nuclear structure, cytoplasmic structure, like crenation of the cell classically caused by enterovirus, or syncytium formation which is caused by measles virus. Another property of the acid production will be there. This property we have already discussed. Adsorption of some guinea pig erythrocyte. If you poured it into the specimen present which is affected with the influenza parainfluenza virus, which have property of hemagglutination or hemadsorption because of the hemagglutinin antigen. So if the cell lines which are affected by virus of influenza, because of influenza the antigen will be present in the host cell which are hemagglutinin, and if you pour guinea pig erythrocytes into that culture tube or cell lines, these will be adsorbed on the affected cells. Another property of virus which can be used for diagnosis of viral affected host cell is growth of non-cytopathogenic virus which inhibit or division of cytopathogenic virus. So growth of first will be inhibit the infection by second. Then property of malignant cell, that is transformation. Property of malignant cell, that is transformation in the cell culture. Then as I said, the virus which incorporate their another last method of cultivation is animal inoculation. That is not commonly used nowadays. The animals used are rabbits, infant suckling mice. Now what changes will occur in the animals? The animal will get diseased, or it may produce some visible lesions on the site of inoculation, or if virus is systemically spread, it can produce lesion all over the body of animal. Then sometimes death of animal can occur. So the diagnosis of virus, here disadvantage is immunity of that animal can hamper the due course of epidemiology or oncogenesis of that particular virus. The serum, the blood can be collected for demonstration of antibodies. Generally paired sera is collected to demonstrate the rise of titer of that particular antibodies. Single serum can be collected for demonstration of IgM antibody. Some rarely used modalities which require stringent precautions, which are labor intensive, require particular trainings, are plaque reduction, neutralization. For practical purposes, in the practical exam, different types of vaccines will be asked to the student, their particular route of administration, their schedule, their dose, their adverse effects. So all these aspects student have to